Hi everybody, this is Christina Howard, and this is a lecture recording for Biology 241. This is Chapter 5 of our Martini Fundamentals of Anatomy and Physiology textbook, and this is skin and its derivatives. So let's begin. I'm going to be using this software called EpicPen to indicate things during the presentation. So let me just test that out really quick by drawing a fun hat on this guy. Perfect, great. Okay, so now we can begin. And to begin with, the skin is the largest organ, and yes, the integumentary system is considered to be an organ. It's about 22 square feet in surface area, and it weighs between 10 and 11 pounds. And obviously this is going to vary depending on how big the human in question is, but on average, those are the figures. So here we have a sort of scary medical illustration of somebody holding up their own skin right here. Oops, and this is the only drawback of EpicPen is that I have to switch between these things. So that's somebody holding up their own skin. So as you can see, medical illustration used to be a lot scarier than it is now. Okay, moving right along. All right, so here we have a little chunk of skin, and the skin or cutaneous membrane contains two major layers. And that is the epidermis, which is a specialized keratinized stratified squamous epithelium. So let's write that down. And I'm going to abbreviate epithelium just for brevity's sake. So that's the tissue type that it is, but as part of the organ system, we call it the epidermis. Epi meaning superficial to, so that's this part of the word, and dermis referring to the layer that is below it, which is the dermis. So this guy right here. So the dermis is below, and the dermis contains the papillary layer, which refers to these little finger-like projections called dermal papillae, and then the reticular layer, which is most of the dermis, and that contains accessory structures like sweat glands, hair follicles, sebaceous glands, the erector pili muscle, and sensory apparatuses like Merkel cells and lamellated corpuscles and tactile corpuscles. How about I just want to move to the next slide. There we go. Okay. So the structure of the skin, as I mentioned, has two layers, epidermis and dermis. And then we have a variety of what we call accessory structures. And the accessory structures are epidermal derivatives. And that just means that they are embryonically derived from the epidermis. So they're not a whole separate thing. They're just a special shape or adaptation of the epidermis. So, functions of the skin and the subcutaneous layer. The subcutaneous layer being the hypodermis, and that's this fatty, very vascular area below here. So one function of the skin and subcutaneous layer is protection from abrasion, shock, and chemical attack. So it helps you basically resist forces that would harm you. It also functions to excrete salts, water, urea, and other organic wastes. But I want to put an asterisk here. These are organic wastes that come from the cells in the immediate area. So the skin and sweating is not a good way to quote-unquote detox. So let's write that. Your liver and your kidneys do that job. So although sweating in, say, a sauna feels nice and invigorating and probably helps clear out your pores, it's not going to help you detoxify from systemic toxins, for example, alcohol or drugs. Those things are dealt with by your liver and kidneys. So just beware of any purchased product that would encourage you to view the skin as a detox organ, because it just straight up isn't. One other function is the maintenance of body temperature. And that actually is performed by the blood vessels in the dermis and hypodermis. So you can move more blood to your body surface to dump heat into your environment. Alternately, you can keep blood away from your body surface to prevent from losing heat. You can also perspire to lose heat via evaporative cooling. That's what sweat is for. Synthesis of vitamin D3 is another one. So we'll talk about the significance of vitamin D later in the PowerPoint, but remember that is another way to say cholecalciferol. So in anatomy, lots of things have more than one name, so this is an example of that. The hypodermis is primarily for storing lipids. As you can see, there's abundant fat at this bottom layer. And then, of course, via the action of sensory structures, we have sensations. So the sensations around touch, heat and cold detection, and pain specifically. Okay, so you've got two types of skin. Thin skin covers most of your body surface, and that's pictured here. So thin really refers to the fact that the stratum corneum is thin. Thick skin, by contrast, has a very thick stratum corneum, as you can see on the picture on the right. So the thin skin covers most of your surfaces, typically has hair, to varying degrees, right? Because you have lots of hair in your head, but less on other parts of your body. There are also sebaceous glands, which are oil glands that lubricate hairs, and they do have sweat glands, but just not very many compared with thick skin. So thick skin is only, I'm going to append only to this bullet point, on palms and soles. It does not have hair, and that's because it needs to grip. It doesn't have any sebaceous glands either, also for grip. Imagine having oily hands and trying to hang on to, say, a tree branch. That wouldn't do you much good, would it? Also, there are numerous sweat glands, again, for grip. So thin films of water allow us to hydrogen bond with things that we pick up, and that helps with gripping as well. So let's move along. The epidermis itself, again, is keratinized stratified squamous epithelium, and we've got 5 kinds of cells here. So these are an important thing to know. So know these. And what I mean by know these is be able to list them and tell me their functions in general, and also which layer you can find them in. In some cases, you'll find a given cell type in more than one layer, and that's okay. But those are the things I need you to know and understand about the kinds of cells that are in the epidermis. And keep in mind, I'm only talking about the kinds of cells that live in here, not the kinds of cells that are down here in the dermis. Those have different functions, and there are different of them. So for a relatively simple tissue like the stratified squamous epithelium to have so many different cell types is pretty interesting, and it's those cell types that give it its function as an organ. So let's talk about these cells one at a time. Come on, there we go. Okay, so basal cells are in the bottom layer that we call the stratum basalis, or stratum basal, or stratum germinativum. Don't worry, you'll see those words again a little later, but that's just this bottom layer of cells here. And these are stem cells that produce keratinocytes above them. So you can see here we have this one, which is in metaphase, as you might recall from 160. And this cell is going to divide, and it's going to produce one cell that remains a stem cell, and one cell that's going to move upward in the column of cells and become a keratinocyte. So because each cell division produces one stem cell and one keratinocyte, you have a constant supply of each of those cells. That's the power of the basal cells. Keratinocytes are most of what you see when you look at the epidermis. So everything from here where I'm circling all the way up to here are keratinocytes, with the exception of this one. Talk about that guy later. these ones are alive, and these ones are dead. So they contain a waterproofing lipid sealant, which they produce while they're still alive. They also produce keratin, which is why they're called keratinocytes. And keratin is the protective component of the dead cells at the top. So it protects from heat, microbes, abrasion, etc. So living cells are pretty fragile, but dead keratin-filled cells are very, very sturdy, and they help keep your outer surface secure. All right, melanocytes. So we'll have a whole unit on human skin color in a little while, but we have to mention the function of melanocytes. So melanocytes in the stratum basal here, which again is this lower layer. So these guys sit in the lower layer, but they have projections upward, which you can actually kind of see the outcome or outline of here. And if you zoom in, you can see the melanocyte likes to sit in the stratum basal, and it puts pseudopods. I'll write that down. Pseudopod means pretend or false foot. So it's the cell version of a foot. And they use those to pass melanin up to the keratinocytes above them. So melanin itself is a pigment that has variable degrees of coloration, so I'll show you the biosynthetic pathway for melanin later, but not all melanin is the same. And this comprises about 8% of epidermal cells. So melanin confers color, but the function of the color is to absorb UV radiation, which protects your DNA and your skin cells from damage. So let's put prevent DNA damage. So that's the function of melanocytes. So we'll talk about human skin color in more detail later, but for now, let's move on to dendritic cells. So I mentioned in lecture that these are perhaps my favorite epidermal cell type. So these are an immune cell, which means that they're kind of looking out for cancer cells and pathogens. So penetrating microbes, anything on the outside that might be trying to get in. And superficial cancers. These are typically rogue keratinocytes that have had some kind of DNA damage suffered, and they are going to start dividing uncontrollably or incorrectly. When they begin to do that, when they're precancerous cells, they begin producing and displaying proteins on their surfaces that are not normal, and the dendritic cell can recognize that and initiate the basically tumor prevention protocol. So the reason that I like dendritic cells so much is because they're kind of like a mall cop. I mentioned this in lecture as well, but it bears repeating. So a mall policeman is capable of affecting change within a limited space and with limited authority. So they can't arrest you or prosecute you. All they can do is grab the offender and take the offender away from the location where the offense is happening. So just like a mall cop catching a shoplifter, they can't arrest that shoplifter, they can't prosecute the shoplifter or charge the shoplifter with a crime. All they can do is drag the shoplifter off to the scary windowless mall office until the actual police arrive. They can do something with the shoplifter, but not the mall cop. Same deal with the dendritic cells. So what they'll do is, let's say this cell right here is going to start dividing inappropriately. So I've drawn a little mischievous face on it. It suffered probably some UV damage from the sun that's damaged its nucleus and its nuclear DNA, and that mutation is causing it to divide uncontrollably, or begin to. The dendritic cell can sense this, and what it's going to do is go engulf the offending cell, and it'll drag the offending cell to the nearest lymph node. So it knows that something is wrong with the cell, but it doesn't know exactly what or how to deal with it. The cells that live in your lymph nodes do. They are much more sophisticated immune cells. So basically the dendritic cell drags the offender off to the nearest lymph node, holds that offender up to the cells in the lymph node and says, hey, I caught this guy doing the bad thing. You deal with it. So they don't have the authority or intelligence to deal with problems directly in themselves, but they can take the offending cell to a place where it can be dealt with. Like little Paul Blarts. If you're not familiar with Paul Blart Mall Cop, I'm sorry that you may miss that reference, but you're welcome to go Google it, of course. Okay, Merkel cells. These guys are down here. So here and here. And these are touch receptors. So they're located in the basal layer. They do have processes that go up. Oopsie, I made a mistake. Sorry. That's what I get for screencasting after a long day of working. There we go. All right, so this is the Merkel cell. How do I know? Well, it's associated with a Merkel disc. It's innervated. It also has cell processes, just not as long. So innervated means that it is associated with a Merkel disc, which is this nerve ending. And that nerve ending carries sensory information away to the central nervous system where it can be interpreted. So these are located in the basal layer of hairless skin. And so these are going to be primarily concentrated on your hands and feet. Okay, so now we're getting into layers, and I want to stress here that layers are groups of cells, and specifically they're groups of, in most cases, non-homogeneous cells, meaning not all the same kind. So when I ask students on exams, list the 5 epidermal cell types, they'll often give me the names of layers instead because there are also 5 of those, but a cell is not a layer and a layer is not a cell. A layer is made out of cells, but it is not itself a cell. So that's a really important distinction to make, and it's one that students often miss or kind of whiff on tests, so I want to make sure that I let you know about that pitfall so that you can try not to make it. Okay, so the stratum basal, which is the very bottom layer, also called the stratum germinativum or the stratum, oops, let's turn that into an M. There we go. It sometimes refers to as the stratum basalis in some texts, so just FYI, that's also acceptable. They are attached to the basement membrane by hemidesmosomes, which is half of a desmosome. And there are multiple cell types in the stratum basal. So the basal cells are the most numerous, and that's because they produce the keratinocytes above. Melanocytes are between 10 and 20%, and this varies across body surface. So the skin tone tends to be darker and more melanocyte rich in the elbows, knees, groin, and axilla or armpit, as well as select other locations, for example, the areola of the nipple. But you're not really the same uniform color across your entire body surface. Merkel cells are sparse and concentrated in the hands and feet. The renewal rate of the epidermis varies by region and by thickness, and the renewal rate really varies by activity of the basal cells, so how fast or slow they are reproducing. They do that by responding to epidermal growth factor, which is a cell signaling molecule. Oops. And that just means it's a receptor binding molecule, so stratum basal cells express receptors for epidermal growth factor, and when they are binding to epidermal growth factor, they are going to be encouraged to divide more frequently. And as I mentioned earlier, this is the embryonic source of the accessory structures of the skin, which we'll cover later. Okay, so the other layers, and this is going from bottom to top, are stratum spinosum. And so this is this. So let's write a 1 next to that. The stratum spinosum is mostly keratinocytes, and it has a few Langerhans cells and a few melanocytes. Most of the melanocytes are in the stratum basalis. This is going to be number 2. Stratum granulosum refers to the fact that the cells here are dying, and they're doing it on purpose, and they're packed with granules of keratohyalin and keratin, which is why they appear speckly in this image. The stratum lucidum is in thick skin only, and if it were present here, it would be right here. The reason it's called lucum is because it's semi-transparent where present. You can see that better on the actual histology slides versus the cutesy little cartoon image we've got here. The stratum corneum is the topmost layer. And these are all dead keratinocytes that are full of lipid waterproofing and also keratin. And they don't have a perfect waterproof. They're water resistant. They're pretty good at it, but as we all know, if you go in the water too long, like you stay in the bathtub too long, you get prune fingers. That's because the waterproofing is not perfect. You also lose about a half a liter of water every day due to what's called insensible perspiration. And this is just evaporation of water from your body surface through your stratum corneum that you have no control over. And this is not sweating that's related to exercise. It's just the amount that you produce and lose to the environment over the course of a day. Because again, waterproofing not perfect. Okay. So now we're finally getting to skin color. So this is melanin up here. And this is garden variety melanin. So a lot of the names for the melanin chemicals have a prefix of some kind, which we'll see in a little bit. So there's more than one kind. So melanin is, let's put an asterisk here, not one size fits all. There are different varieties. Keratin is another one of the epidermal pigments, and it looks like this. So this is an orangish pigment. Melanin ranges from yellow, orange to black. The other thing that gives your skin its color is hemoglobin, which is right here. So hemoglobin carries oxygen in your blood. Specifically, the oxygen binds to this iron ion right here in the middle. And when it does that, it reflects red light. So it gives skin a reddish pink color. So that, combined with the amounts of melanin and keratin and what kind of melanin you have, really can make a super diverse variety of skin tones in humans, which is pretty cool. So here's keratin, and keratin gives an orange coloration. So it's yellowish orange. You find it in, for example, vegetables with lots of keratin. So carrots, for example, pumpkin, squash, I could go on. Anything you can think of that's orange and is a vegetable and you eat it, very rich in beta carotene. Also, the reason that, and let's, to be festive, let's make my pen orange. So it accumulates in the stratum corneum and in the subcutaneous fat. So human fat is yellowish orange. Oops. And that is because of the accumulation of beta carotene. So I'm going to pause here. And I'm going to pause. Okay, hopefully we're recording again. So sorry about that. My computer is running low on batteries and I had to go back and get it up and running. So let me just go down here And we're going to go through these. And I want restore. All right, so now we're going to move on. So yes, it accumulates in the stratum corneum and in the subcutaneous fat. And that is actually a good thing because that allows you to store your vitamin A because it's a precursor to vitamin A. Vitamin A is required for proper vision. So the pigment molecule in your retinas that changes shape in response to light and makes your retinas work is built using beta carotene as the starting material. So if you don't have a steady supply of it, you can't replace that pigment. It's called the visual pigment. And it's also required for epithelium maintenance, so it's going to help make sure that your cells are turning over at the appropriate rate, which is also very critical. So you need enough beta carotene, not too much. Too much can be really bad because it can cause too much vitamin A, which leads to symptoms like joint pain, etc. But that's really a vitamin A thing more than it is a beta carotene thing. Okay, so I promised I would show you the melanin pathway and explain how there's different kinds of melanin, and now I will. But I do want to point out this biosynthetic pathway that's shown here, you do not have to memorize that. It's too complicated and beyond the scope of this class to memorize that. I'm just showing you where the different kinds of melanin come from. So that's the idea here. So the main idea of melanin is to protect from UV radiation. So earlier I showed you the little melanocyte passing its melanosomes up towards the keratinocytes above it. And melanin actually varies in pigment from pale yellow to black. So the pale yellow melanins are in the pheomelanins family. The eumelanins are the darker ones. So pheomelanins are yellow to, oops, momentarily lost the ability to spell. There we go, yellow to orange. And the eumelanins are brown to black. The precursor material for all melanin, regardless of what color it manifests as, is the amino acid tyrosine. An enzyme called tyrosinase converts tyrosine into dopa and then dopaquinone. And then from here, you have two pathway options. You can either go down the route to eumelanins, or you can add a cysteine and go down the route to pheomelanins. Now, note that there's an S on each of these. That means that actually eumelanins, there's a group of them. We're not going to get into which are which, other than to say eumelanins and pheomelanins are the metabolic outcomes of different pathways. And it doesn't mean that you only have a eumelanin pathway or only have a pheomelanin. Most individuals express some amount of both, but the relative amounts of eumelanins to pheomelanins, along with carotene and with hemoglobin, are going to be what determines your skin color. So this individual here is obviously a lot paler than the people he's with. These are actually his grandparents. So you can see that there's a significant difference in coloration. His grandparents are very dark-skinned individuals, and he is a very light-skinned individual. So if they're related, what accounts for the skin difference? This person has a form of albinism, and albinism in this form is a congenital defect, so congenital meaning heritable, a defect in the gene that codes for tyrosinase. So they have either tyrosinase that is entirely non-functional or that is not even made, which means that even if they have enough tyrosine, they can't initiate the pathway to either eumelanins or pheomelanins, and that means they can't make pigment at all. So these individuals are very fair-skinned. They have pink irises because there's no melanin in their iris. That makes their vision very sensitive to light, and they have increased risk of melanoma because they don't have any protective pigments in their skin. So to explain how UV radiation is blocked by melanin, I'm going to get rid of all this, and we're going to switch over to whiteboard mode for a moment. It's one of the cool things about this program that I have here is that you can do that. So let me draw my melanocyte. And then I'll draw some, oopsie, little pigment vesicles inside of some of the keratinocytes above. So, melanosomes, melano means black, and somes means body. So these are just little vesicles full of melanin pigments. So what happens is the melanosomes get transported up, 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 and they enter the keratinocytes. So what the keratinocyte is going to do is it's going to position the melanosomes on the side of its nucleus that faces UV radiation. So let's draw myself a sun up here, a rudimentary childlike sun though it may be, and UV radiation is going to come down and it's going to make its way through the stratum corneum and end up hitting a living keratinocyte. We don't want that UV radiation to hit the DNA in the nucleus. That would be bad. It might damage the DNA in a way that would be harmful. Fortunately, melanin readily absorbs that radiation and prevents the radiation from affecting the DNA. So that's the point of having melanosomes. It's actually a little bitty molecular umbrella. Which is pretty neat. Okay, so that's the basics of how melanosomes are utilized by keratinocytes. And no more whiteboard. Okay, so now that you get the idea, you understand more about how melanin is used and why it's produced. So let's move along and account for differences in skin color. Oh, by the way, the tyrosinase thing was just for your information, so I'm not going to test you directly on it. I'm just explaining to you that disruption of one enzyme in a biosynthetic pathway can produce pretty significant results for phenotype or outcome. So as I mentioned in my whiteboard drawing, melanosomes are vesicles that contain melanin, and these are taken up by keratinocytes. So that shields the keratinocyte from UV rays and decreases UV damage to DNA. Earlier, I mentioned that not all of your body surface has the same number of melanocytes, which means that not all of your body surface is the same color. So in your fairer skin across your body surface, it's about 1 in every 20 cells. That's a melanocyte in your skin. And in darker skin, it's only about 1 in every 4. So what accounts for differences in skin color? Well, the number of melanocytes is not it. So if you count melanocytes in skin over body surface in individuals of many different colors, the number of melanocytes that is possessed by individuals is consistent across races. So it's not that. In fact, there's a lot of things that play into human skin color differences, not just one. So one is variation in color of melanin, so how much pheomelanin you produce versus how much eumelanin. The other is differential rates of melanin synthesis. So some individuals produce more melanin more quickly than others. The variable dispersion of melanocyte or melanosomes in keratinocytes. So maybe one clump of keratinocytes possesses more melanosomes than their neighbors. So people that have persistent year-round freckles, they have sort of a variable dispersion. The clumping and the size of the melanosomes as well. And importantly, the differences in the rate of melanin hydrolysis by lysosomes. So in keratinocytes that are receiving melanin that they're not going to use, instead of positioning the melanin on the top of their nucleus in the manner I described previously, they just go ahead and fuse it with a lysosome and get rid of it because they don't need it. So all of those things are heritable, which is why we get our skin colors from our parents and from our ancestors. So UV exposure increases the rate of melanin production. So this is what accounts for things like tanning, right? So if I spend a lot of time in the sun, even though I'm a very pale, pasty individual, I'm probably going to get darker over time because my UV exposure is up, and so my melanin production also needs to rise. It also increases the mitotic rate of the stratum germinativum. So it's a bit of a double-edged sword because the more mitosis a tissue does, the likelier it is that there will be a replication error in the DNA that will produce cancer. But high cell turnover is also good. So these two things go up at the same time so that bullet point 1 can protect bullet point 2. So this begs the question, everybody experiences sunlight, and if melanin is so protective, then why aren't all humans dark skinned? And the answer is twofold. One, evolution is not perfecting. It only acts on the genes or gene pool present, and it can only modify stuff that already exists. So it's not a perfecting process. It doesn't make ultra mega humans. It just changes people gradually over evolutionary time so that they are adapted to their environment a little bit better incrementally. The other is, of course, that different areas of the globe receive radically different amounts of sunlight throughout the year, and the angle of the sunlight also affects the sun's intensity. So at the equator, sunlight is hitting the Earth's surface at an almost 90-degree angle, which makes its intensity very, very high. Also at the equator, it's sunny for most of the year, versus at the poles, it's literally dark for half the year. So there's going to be lots of variation according to latitude as far as how much light a population of humans is getting. So that's why people from very, very northern latitudes tend to be paler, and people from lower latitudes closer to the equator tend to be darker.